Hello, my name is Daniel Chichinsky. I'm a graduate student in the Land Resources and Environmental Sciences Department at Montana State University. This project is on the integration of thistle rust into organic management of Canada thistle. I'd like to thank my advisors, committee members, mentors, and lab technicians who have all helped get this project underway. I would also like to thank Western SARE for their contribution to this study, as well as the Montana Organic Association. Cirsium arvense is also called Canada thistle, California thistle, or creeping thistle. It is a perennial invasive weed that is found throughout temperate climates of the world. It is common in Montana and in the northern Great Plains. This weed reproduces through the production of germinating seed as well as clonal rhizome. And clonal rhizome are basically a network of creeping roots which can produce new shoots that have the ability to sexually reproduce and disperse seed into new habitat. Canada thistle has the potential to lead to serious crop yield loss, particularly in organic systems that don't have the option of using synthetic herbicides. They can also reduce diversity in native and natural systems. Typical controls for Canada thistle include herbicide application, competitive cropping, mowing, burning, grazing, and cultivation. So, why does Canada thistle matter and what are we doing about it? In 2011, the Montana Organic Advisory and Education Council surveyed organic farmers in Montana, just asking them what their research needs were, and they responded with the need for more information on organic management of perennial weeds. In particular, Canada thistle was a primary weed of concern. So, weed ecologists, agronomists, and researchers at MSU responded by performing a meta-analysis where they compared 100 peer-reviewed studies which focused on the management of Canada thistle, and there were two key takeaways. First, the majority of publications focused on synthetic herbicide-based management of Canada thistle, with very little research on non-chemical management. And second, they compared the reported management impacts on Canada thistle across all of these different studies and found that studies which had integrated two or more management techniques also had the highest reductions in Canada thistle. So again, we need more research on non-chemical management of perennial weeds, and integrated management is definitely the best overall way to reduce Canada thistle in both organic and conventional systems. So integrated weed management is a combination of weed management tools that reduces the negative consequences of singular management approaches. In general, chemical management isn't in the organic toolbox. However, it's important to know that chemical weed management works, but over-reliance on synthetic herbicides is really accelerating weed adaptation and resistance, only stressing the need for integrated, multifaceted management approaches. Typically, organic producers utilize mechanical, cultural, and biological tool sets. Mechanical management is the most common approach, but when used repeatedly over time, mechanical management can have some negative impacts, especially in terms of soil health. Additionally, the act of breaking Canada thistle rhizome with tillage implements really only spreads the clonal rhizome to new habitat and causes an increase in the thistle population. And this goes back to the biology and reproductive capability of the thistle rhizome. Cultural management is based on the use of things like diversified cropping systems, utilizing competitive crop varieties, incorporating narrow row spacing, etc. These techniques may be low impact alone, but can be really powerful when they're added to an integrated weed management approach. And last but not least, biological management is thought of as an approach that uses living beneficial organisms, such as grazing animals, or perhaps considers the use of buffer strips that act as habitat for beneficial insects. However, the act of using beneficial pathogens as a biological weed control is still a pretty novel approach. So that leads me to Puccinia punctiformis. It's also called thistle rust. Thistle rust is from the Basidiomycota family of fungi and has become naturalized to all ranges where Canada thistle exists. It is highly selective to Canada thistle and has no alternative hosts, making it a safe and reliable option. Thistle rust acts as an obligate parasite where it needs living host tissue to complete its life cycle. The fungus lives off of sugars within the Canada thistle rhizome, and when it's ready to reproduce, it emerges as spores on above ground vegetation. If the mature spores don't immediately come in contact with a new host, they can remain dormant within Canada thistle leaf debris until they come in contact with the thistle rosette. Generally, successful infection requires mild temperatures and the presence of moisture. So, our tillage study, or excuse me, so we have actually actively been conducting research over the last 2 years, and mainly our goal is to explore the potential to integrate thistle rust as a Canada thistle biocontrol within organic agroecosystems. Our focus is on thistle rust response to agronomic practices. Our first question is focusing on tillage. So does tillage, conventional versus reduced, help? Does it hurt or have no impact on the spread and establishment of thistle rust? Second, we are looking into cropping rotations. So we want to know is does thistle rust cause Canada thistle patches to decline at higher rates when grown with competitive crops in a diversified rotation? And then third, we are looking at extension work. So we want to work with farmers and stakeholders to build functional integrated weed management systems. Okay, that leads me to our tillage study, which is occurring at the Fort Ellis Research Farm in Bozeman, Montana. This study was started in 2020 and will finish in 2022. These maps are showing the changes in our experimental fields over the last 2 summers. Each year, we collected Canada thistle and thistle rust growth data from 4 fields. These fields were randomly split into a reduced tillage mowing treatment or a standard tillage disc cultivation treatment. We want to understand how thistle rust and Canada thistle abundance is impacted by these tillage practices. So what we're doing is we map the perimeter of each discrete thistle patch with an EMLID GPS receiver. We also take random samples of Canada thistle density from within each patch, and we do a complete census of Canada thistle stems, which show thistle rust symptoms. And these maps are showing the rate of symptomatically infected Canada thistle within each patch between the years 2020 and 2021. The main differences between the years are that a lot of the patches actually merged in 2021, but overall thistle density declined. Additionally, the abundance of the thistle rust pathogen has increased between 2020 and 2021. This figure shows how the Canada thistle stem abundance has changed over the 2 years of the study. The colors represent thistle patches that were either completely asymptomatic with no disease symptoms observed, which is the teal color, or patches that had thistle rust symptoms to varying degrees, and that's the red color. Additionally, we can compare the effects of the tillage treatments between years. So first of all, Canada thistle density was reduced between 2020 and 2021. On average, Canada thistle abundance was reduced by 53% in the disc treatment and by 55% in the mow treatment. There was a higher reduction in patches that were asymptomatic of thistle rust compared to patches which had no disease symptoms. And this phenomenon is actually consistent with what's happening in our other studies. We're seeing that the pathogen is more likely to be found in high density Canada thistle patches. And this is really a window into the ecology of the Thistle Rust pathogen, showing us that it's just better to exist where there's an abundance of resources to live and reproduce off of. As a quick disclaimer, I think it's important to note that we have been experiencing low moisture conditions at the Fort Ellis Research Farm over the last couple of years. So it's necessary to assume that this reduction in thistle density can also be attributed to lack of water. Now this figure is showing the change in abundance of symptomatically infected Canada thistle stems over the last 2 years. We have each year split by tillage treatments, and what we're seeing is that the thistle rust abundance has increased by 61% in the disc treatment and by 51% in the mow treatment. And this is a pretty good sign. It's basically showing us that our efforts are actually facilitating an increase in this beneficial pathogen. And that hopefully in years following, we will have an increase in the abundance of transmissible spores that exist on our soil surface and which can eventually parasitize new Canada thistle hosts. So it will really be pretty interesting to see how this affects Canada thistle growth over the last year of this study. I am also testing the effects of a competitive rotation at the greenhouse at MSU Bozeman. So Canada thistle is grown perennially in 10-inch pots where we simulate 4 perennial growth cycles. There are two treatments, each isolated in their own climate-controlled rooms to prevent unwanted movement of spores. We have our control, which hasn't been inoculated or exposed to thistle rust, represented by the blue pots in this image. And we have a treatment, where Candida thistle is repeatedly inoculated with the pathogen, represented by the red pots in this image. I'm also testing and comparing the competitive effects of a four-phase diversified rotation between inoculated and control treatments, where I'm comparing the growth of a specific crop in monoculture. I'm looking at the growth of Candida thistle in monoculture, and I'm looking at the growth of Candida thistle in a crop when they're grown together in polyculture. So our experimental rotation includes four phases that are being repeated over three separate trials. Each phase is harvested at crop maturity, and the perennial Candida thistle is allowed to regrow from its clonal rhizome. I also inoculate the thistle rust treatment once thistle has regrown into the rosette stage, adding up to a total of 4 different inoculations over the entire rotation. Growth data is collected at the time of harvest, where I focus on abundance and biomass of the crop and the weed, as well as abundance of symptomatically infected thistle stems. After harvest, I directly sow seeds for the next rotational phase into the pots using USDA recommended organic seeding rates. So our 4 phases include phase 1, which is a simulated fallow, which is basically where we allow Candida thistle rhizome to establish without any competition. Phase 2 is a competitive treatment of hard red spring wheat, followed by Arvica forage peas in phase 3. And phase 4 is a safflower variety that we acquired from the Central Ag Research Station. To evaluate the effectiveness of competition between the control and inoculated treatments in the greenhouse, we are using something called the Relative Competition Index. In this index, we basically assume that any plant which is grown in monoculture will reach a maximum growth potential if all of its needs are met and held constant throughout the trial. In contrast, we assume that a plant which is grown in direct competition with another species will have reduced growth. And that's because now it's forced to compete for space, light, moisture, and nutrients. And we're actually seeing those differences when we look at the biomass of Candida thistle when grown alone in monoculture versus in direct competition with wheat, peas, and safflower. So really my goal is to see what happens after we add a disease that specifically attacks one species in the system, being Candida thistle, with the expectation that thistle rust will reduce thistle's competitive ability. So RCI here on the far right corner of this figure is basically the percent weight reduction of Candida thistle between thistle grown in monoculture and thistle grown in polyculture. And so in trial 1, it turned out that thistle biomass was actually reduced more in our non-inoculated control when it was in competition with wheat and peas. But by the time safflower was harvested, there was a switch. Now the effects of competition favors the inoculated treatment, resulting in a larger weight reduction of our thistle. And there may be some reasons for this. By this time in the trial, thistle had been harvested multiple times, and it was under competitive stress from 3 separate crops. Additionally, the thistle rust pathogen had nearly a full year to establish in the rhizome, a year to produce spores, and really begin to start taking effect as a thistle parasite. So we can begin to think that maybe this pathogen just takes a little while before it really becomes an effective thistle parasite. But there's still time to work through these nuances. We are in our second trial, which is scheduled to be completed in a few weeks, and the third and final trial will come to a close by next summer. And by then, we hope to have some clear data-driven answers as to the effects of thistle rust in competitive systems. So just to summarize, there will be one more year before all of these studies come to a close. At this time, we can say for sure that thistle rust does have a role to play as a Candida thistle biocontrol. While it's not the hole-in-one answer to the Candida thistle problem, it's definitely a low-cost and safe addition to the integrated management toolset. Thank you.